Welcome back, students. This is Module 4.1.4, DNA replication. DNA replication may be defined as the process by which DNA copies itself in preparation for cell division. This is the basis of heredity, as reproduction is done by cell division, and DNA replication duplicates the DNA in preparation for cell division. So this is a process that really drives inheritance and heredity. Now, the way in which DNA replicates is known as semi-conservative replication. So you have a double-stranded DNA molecule, that twisted ladder, and what happens in semi-conservative replication is that it unwinds, and each half of the ladder serves as a template. You recall that a template is something that is copied. And so each of the new double helixes is half new and half old. So let's go ahead and look at that next slide right now. As you can see here, the old DNA is blue, and when it replicates, each of the old halves of the blue DNA serves as a template and is copied. And you can see then that this is what results from semi-conservative replication, that each of the new strands is half new and half old, if you look in the illustration here on the right. So let's go back where we were. DNA replication, as you already know, occurs in S phase of interphase. As a matter of fact, this little illustration here in the lower right that shows homologous chromosomes, a purple one from mom and a green one from dad, same size and same centromere location, that when they replicate, then there's twice as much DNA, but because the centromere didn't duplicate or divide, the number of chromosomes didn't change. And so we can see that the difference between unreplicated chromosomes on the right and replicated chromosomes on the right, I hope I said that right, unreplicated chromosomes on the left and replicated chromosomes on the right, the difference is S phase of interphase and DNA replication. And that's an important principle for you to recognize, and it ties it to what you already know about mitotic cell division, for example. In this illustration we've already looked at, you should be able to understand and explain what is meant by semi-conservative replication. The process of replication itself is fairly straightforward. It has several steps that you should be familiar with. You should be able to recount these steps and explain what happens during replication. The version that I give you here in Biology 101 is a little less detail heavy than if you were in Biology 181, the class for biology majors, which only makes sense. But these are simplified steps, is what I'm saying. The first step is unwinding. Notice that I simply say that the two strands of the double helix unwind with the help of an enzyme. I'm not making you learn the name of the enzyme. And when I say H bonds, I mean hydrogen bonds between bases. You'll recall that hydrogen bonds connect the bases of the two halves of the DNA molecule. And so the H bonds are broken. Sometimes I say they're unzipped. And this allows the information inside, the order of the bases, to be used, to be copied. The second step takes advantage of the exposed bases, and the second step is known as complementary base pairing. So each of the now exposed bases is used to bring its complement in, and this is what dictates the order of the nucleotides, the free nucleotides, as they come in. A free nucleotide is just a nucleotide that's not connected to a chain of DNA or RNA. And so they join to their complement. And remember, complementary base pairing, A is always with T, G is always with C, adenine with thymine, guanine with cytosine. And so that's what determines the order. And so you can see complementary base pairing is critically important, because otherwise, if you get things out of order, if complementary base pairing isn't carried out, then you have a mutation, a change in the sequence. Now, just putting the nucleotides in order isn't enough. They must be covalently bonded there, and those bonds, with this process of covalent bonding, we call joining. And here is the name of an enzyme I would like you to know and associate with this process of replication. The enzyme which kind of controls replication is called DNA polymerase, and I think that's very well named because it's making a DNA polymer, and then it just has the enzyme ending ASE. So DNA polymerase makes a DNA polymer. That's what's happening here in DNA replication, is we're making another DNA molecule. So then it will covalently bond, attach those complementary bases to the 3' prime end, and so replication only proceeds from the 5' to the 3' end. And as those bases are put on, the molecule rewinds. Now, DNA polymerase has an additional ability. It is able to proofread. It compares the bases that are placed next to the template bases and make sure that they are the correct ones. And if an error is found, it can actually repair and put the correct base there. And so the result of having this proofreading function of DNA polymerase is that the majority of errors are corrected. This keeps the mutation rate very low. An uncorrected error in DNA replication results in a mutation. And so there we go. I think in order to really appreciate this process, you should Google replication animation and watch a couple. Here's one. It's not especially good. You have to click to go through it, but it's okay. It's so you can watch that one or find one of your own to look at. Here's Mr. and Mrs. DNA, Mr. and Ms. DNA at a shishi restaurant, and she leans over the table and says, "Psst, darling, your helix is unzipped," and that's that pesky enzyme that unzips it. Here's an illustration of what the process of DNA replication looks like in a very simplified fashion. This doesn't even show that DNA is helical, that is a twisted ladder. It just shows it as a ladder. It doesn't really show anything except the bases. The backbones here, which you will recall are of sugar and phosphate groups alternating, are there. But in any case, so you unzip and unwind, and so when it's unzipped, you can have this information available, the order of the bases, and these little individual nucleotides here with a G and a T and an ASC. These are free nucleotides, and they just pair up according to the order of the nucleotides, the bases and the nucleotides of the template. And that's basically the process of replication. There's more to it. If you were in a class for majors, would tell you more detail, but that's sufficient for our purposes here. And that will conclude this module.